Hello children, now we are starting chapter number 2 of your textbook Curiosity, and the name of your chapter is The Invisible Living World Beyond Our Naked Eyes. So children, please share your questions here. Have you ever wondered what you might see if the invisible world around you become visible? So what you might see? How do you think your observation of this hidden world might change the way you think about size, complexity, or even what counts as living? So, how your observation will be changed? And have you thought how these tiny living beings interact with each other? Even we cannot see them, so please think that how they can interact with each other. Now share your question and we start the chapter. The human eye can only see objects that are above a certain size, okay. So, the objects of a certain size we can see, otherwise there are so many invisible things in the world which we cannot see with our naked eyes. For a long time, many tiny things around us remained unknown. Long ago, people discovered that a curved piece of glass could make small things look bigger, right? You must have seen that amplifying glass. So, the piece of glass was shaped like a lentil seed, thick in the middle and thin at the edge. So, that was thick in the middle and thin at the edges. Hence, they called it a lens. Over time, lenses were improved to become more powerful. Each new tool, from simple magnifying glasses to microscopes, helped humans see what their eyes could not. So children, by the time first of all lenses were invented, then magnifying glasses to microscopes, this journey was very long but very helpful in seeing the things which human eyes cannot. The invention of the microscope opened a fascinating hidden world filled with tiny living creatures. We will explore some of these life forms in this chapter. You have already learned about the amazing variety of living beings, right? Just look around. There are so many beautiful plants and animals which you can see obviously. They are of all shapes, size and colors. Some living beings are tiny while others are really big. They differ not only in their structure but also in many other features, right? Their structure is different, their survival is different. Now, all these living beings, whether plants or animals, are called organisms. Have you ever noticed the smallest organism around you that is visible to the naked eye? Think about it, how small a thing can your eyes actually see? You might have seen some people using reading glasses. How does it help them see better? Or what happens when we use a magnifying glass to observe something? So, let's do our first activity of this chapter. Take a round bottom flask made up of glass as shown in figure 2.1. Fill it with water. Okay. So, you have to take a round bottom flask which is made up of glass, and now you will fill it with water. Close the mouth of the flask with a cork. Now, place the flask on an open book. Okay, like this picture, and look at the letters through it. Now, you are looking the letters through it. Do you notice something interesting, children? The letters appear larger when seen through the flask, right? You can see in this figure. This happens because the flask filled with water acts like a magnifying glass. Okay, now the flask is doing the work like a magnifying glass. Now use a real magnifying glass look at small organisms like an ant? Were you able to see the details of its body more clearly? Obviously, you will be. For a long time, people were curious to explore the tiny organisms around them, but they could not see them with their naked eyes. So how did we finally discover this invisible world with the help of these tools? Do you know which scientific discovery helped us see the tiny world for the first time? Let's know about it. So, in 1665, a scientist named Robert Hooke published a book called Micrographia. Okay, what was the name of this book? Micrographia. He was a careful observer and a skilled artist, right? In this book, he showed detailed drawings of tiny things that people had never seen before. Things he saw using a tool we now call a microscope. His microscope made things look 200 to 300 times bigger than what one could see with the unaided eye. One day, he looked at a thin slice of cork and saw it was made of many small empty spaces. These compartments reminded him of a honeycomb. He drew what he saw, okay, and called each small space a cell, okay. So, he gave the name cell to each small space. This was the first time the word cell was used in science to describe the basic unit of life. Around the same time in 1660s, Antony van Leeuwenhoek, a Dutch scientist, made better lenses, okay, that allowed him to build more useful microscopes. He was the first person to clearly see and describe tiny living things like bacteria and blood cells. Because of this, he is known as the father of microbiology. So now you have this cover page of the book here, Micrographia, or some physiological description of minute bodies made by magnifying glass with observation and inquiries thereupon. Okay, so just go through with that. Now we have Micrographia book, figure 2.2. Okay, this is microscope of Robert Hooke and this is cork cells published in Micrographia. Now children, what is a cell? All living beings are made up of cells, right? You might wonder what cells actually look like. Have you ever seen a cell? Let us take a closer look at the basic structure of a cell using a microscope. Now we have activity 2.2. Let us study a cell. Take an onion bulb from your kitchen like this or garden and wash it thoroughly with water. Cut the onion bulb vertically into pieces. Take one piece of onion and pull out the thin transparent layer from its inner surface with the help of forceps. This layer is called the onion peel. Place the peel in the petri dish containing a few drops of safranin, that is red colored stain, for 30 seconds. Okay, you will place that peel in a dish containing a few drops of a red colored stain. This will give a pinkish color to the cells and help us see them clearly. With the help of thin brush, transfer the onion peel to another petri dish. Okay, now you will transfer it containing water to rinse the peel and remove extra stain. Now carefully place the stained onion peel on the glass slide using a thin brush. Now we are placing it on a glass slide, ensuring it does not break or fold. Put a drop of glycerine, okay, you can see the picture number 4 here. Put a drop of glycerine over the onion peel on the slide. The glycerine will prevent drying of the cells and improve clarity for better visualization of cells. Slowly place a cover slip like this. This is cover slip over the peel using a needle such that no air bubbles get trapped. Use blotting paper to gently wipe off any extra glycerine around the edges of the cover slip. Observe the slide under a microscope. A simple foldable paper microscope will suffice. Compare it with figure 2.3c. Okay. So, this is cell wall, cell membrane, nucleus and cytoplasm. How we are able to see it? Through a microscope. What similarities do you find in figure 2.3c and figure 2.3d? You will observe nearly rectangular structure under the microscope. These are the cells of the onion peel, which are closely arranged without any space between them. Try to observe the peels of the leaves of different plants around you. You will find that all plants are made up of cells. What do you think the body of an animal is made of? Let's see. Now we have activity 2.3. Rinse your mouth with clean water. Use the blunt end of a clean toothpick and gently scrap the inside of your cheek. Place the scrapped material in a drop of water on a clean glass slide and spread it evenly. Add a drop of methylene blue, a blue colored stain, over the material on slide. Adding stain improves the visibility of the material under the microscope by increasing contrast. Okay. So, when we add the stain, so what happens? It improves the visibility of the material. After 1 minute, add a drop of glycerine over the material on the slide to prevent the cells from drying. Now, carefully place a clean cover slip on the material, right, and remove the excess glycerine from the edges of the cover slip using blotting paper, just like our previous experiment. Observe the slide under a microscope and draw what you see in your notebook. Okay. What did you observe, children? You will observe a polygon shaped structure as shown in figure 2.4. Now, these are cheek cells which form the inner lining of your mouth. What similarities and differences did you observe between the cells of onion peel in activity 2.2 and human cheek cells in activity 2.3? Can you tell me? You have observed that cells have three main parts, right? You can see here three main parts. The outer layer is called the cell membrane. The round structure in the middle is called the nucleus, which is also covered by a thin membrane. The space between the cell membrane and nucleus is filled with cytoplasm. These three, cell membrane, cytoplasm and nucleus, are the basic part of a cell. What are the basic part? Cell membrane, cytoplasm and nucleus. Now, some cells like onion peel cells have an extra outer layer called the cell wall. What is the importance of these structures in a cell? What functions do they perform? Are these functions important for the maintenance of life? So, now some questions are occurring in our mind after doing this experiment. This is science, children. First of all, we observe something, we notice something, then we do experiment on those things, and then some more questions occur in our mind. The cell membrane encloses the cytoplasm and nucleus. The cell membrane separates one cell from another. What is the work of cell membrane? It separates one cell from another. It is porous and allows the entry of materials essential for life processes and the exit of waste material. Cytoplasm contains other components of the cell and compounds such as carbohydrates, proteins, fats and mineral salts. Most of the life processes take place within the cytoplasm because it contains what? Important compounds of the cell like carbohydrates, proteins, minerals. The nucleus regulates all activities that occur within the cells. It also regulates growth. The cell wall in the plant cell provides rigidity and strength to the plant. Okay. So, what cell wall does in plants? It gives rigidity and strength. This is why all cells are arranged compactly with each other and look firm in structure. So, cells in all parts of a plant have tiny rod shaped structure called plastids. Some plastids like chloroplasts contains chlorophyll, okay, which makes them green and helps in photosynthesis. So, what makes them green? Chloroplast, which contain chlorophyll. In non-green plants which are not green, they help in the storage of substances. Plant cells also have a large empty looking space called a vacuole. This helps the plant cell stores important substances, get rid of waste and maintain the shapes of the cell. This gives strength and support to the plant. In animal cells, vacuoles are usually not present. If present, they are usually small. These small vacuoles stores certain substances dissolved in water. Figure 2.5 you can see, children. So, a cell is not just a simple bag of liquid. It is a complex structure made up of many different parts, each with its own special function to allow the cell and in turn the entire organisms to work. Okay. So, how a tiny cell is so important, which we know now through a microscope, with the help of a microscope. This is figure 2.5, a schematic drawing of an animal cell and a plant cell. This is an animal cell, this is a plant cell. The colors are to show different parts of the cell. We have now understood the basic structure of cell, right? And we now also understand that plant and animal cells differ in shape and structure. Now we have one more question, children. Do different animal cells also vary in their shapes and structure? Variation in shape and structures of cell. Let's read it. The muscle cell and the nerve cell of a human are shown in figure 2.6. Okay, A and B. This is muscle cell, this is nerve cell. What are the similarities and differences you see in them? A muscle cell is shaped like a spindle, while a nerve cell is very long and has branches. You can see the branches here. Similarly, some cells are round in shape while others are long and thin. The number of cells also varies in different organisms. Why do cells look so different from each other? Does the shape and structure of a cell relate to its function? Let's know about it. The unique shape, size and structure of cells help them carry out their specific functions. Okay? We carry out our specific functions due to our cells. But how do these cells help in performing various functions in the body? Let us find out. You observed in activity 2.3 that inner cheek cells are thin and flat, right? They form a protective lining on the inner surface of the cheek. Nerve cells, also known as neurons, carry messages in our body. The elongated shape and branch structure help them reach different parts of the body and pass on messages quickly. Okay? So, you have seen that in nerve cells it has elongated shape and branch structure. So, what it does? It helps them to reach different parts of the body and pass on messages quickly. Similarly, plant cells also show variations. In plants too, cells may be rectangular, elongated, oval or even tube-like, right? Some plant cells form long tubes that help carry water throughout the plant. You have already studied the digestive system in grade 7, right children? Different parts of the digestive systems are made up of different types of cells. Okay? A group of muscle cells are present in the food pipe. Now these cells contract and relax in a wave-like manner, pushing the food down to the stomach. If muscle cells are not thin, flexible and spindle shaped, it would not be possible. What? To push the food down to the stomach. The stomach also has different types of cells for performing different functions. Muscle cells in the stomach wall help churn the food. Other cells in the inner lining of the stomach produce digestive juices and acid that help break down the food. All these cells work together to make digestion possible. Now we will understand children that what are the levels of organization in the body of a living organisms. The body of a living organism is organized in a complex way. Cell is the basic unit of life, right? Just like a brick is the basic unit of wall. A group of similar cells forms a type of tissue. Okay? Different tissues are organized to form an organ. You can see here different tissues are organized to form an organ. Several organs work together to form an organ system. This is an organ system that performs some major functions of the body. All the organ system together make a complete organism like a plant or an animal. So, the levels of organizations are first of all cell, tissue, organ system and then organism. These levels of organization help us understand how simple building blocks like cells come together to form a complex living being. The life of complex living organisms begins with a single cell egg. Okay? The egg of any organism has an amazing ability, okay, to divide repeatedly to form a complete living being made up of many cells. Such living beings are called multicellular organisms. Animals including humans and plants are all examples of multicellular organisms. Have you ever heard of the yolk, the yellow part of an egg? Okay, you must have eaten it. Of an ostrich egg is a single cell. Okay, so the yolk of an ostrich egg is a single cell, the largest known cell in the living world, like this, measuring about 130 mm to 170 mm in diameter. The egg contains extra non-cellular material, a shell for protection and a white liquid that nourishes the cell during its continued development. Okay. So, the largest known cell is the yolk of an ostrich egg is a single cell, and that is the largest known cell in the living world. We have given the measurement here also. So, now children, we will see what are microorganisms. Some living organisms are made up of just one or very few cells. Okay? They are so small that they cannot be seen with the naked eye. These are called microorganisms. Some microorganisms like bacteria and amoeba are made of just one cell. Okay? So, these are unicellular. Others like some fungi and algae have many cells, multicellular. They are found all around us in water, soil, air and even inside our body. But what do their cells look like? Are they like the plant and animal cells we just learnt about, or are they different? Okay, now these questions are coming in our mind. To observe the cells of a microorganism, again we need to use a microscope which magnifies their sizes and makes them visible to us. Scientists have also created a low cost and foldable paper microscope. Such foldable paper microscopes may not provide the same level of details like high powered laboratory microscopes. Okay? However, these make microscopic world accessible to many people. Let us now take a closer look at the fascinating world of microbes. So, we have now activity 2.4 to observe pond water, stagnant water. Okay? Take a container and collect pond or stagnant water in it with the help of your teacher or elders. Use a dropper and place a drop of pond or stagnant water on a microscope slide. Put a cover slip and observe it under the microscope. Observe the tiny organisms found in the pond or stagnant water. We have another activity to observe soil suspension. Take a beaker, children, and collect some moist soil in it from the nearby field or garden. Not dry soil, moist soil. Do not touch the soil with your bare hands. Use a spoon or gloves. Pour some water into the beaker and stir it with a glass rod. Okay? The liquid, and which may look dirty, has very fine particles of soil and is called soil suspension. Keep it aside for some time and let the mixture settle. Use a dropper and take a drop of water from the top layer. Place the drop on a microscope slide. Cover it gently with the cover slip and observe it under the microscope. You may observe small moving organisms similar to those you saw in activity 2.4. This indicates that even soil suspension contains a variety of tiny creatures that cannot be seen with the unaided eye. The tiny creatures that cannot be seen with the naked eyes are called microorganisms. Micro means very small, organisms means living beings or microbes. We have another activity. Let us study. A group of students studying in grade 8 performed activities 2.4 and 2.5. They also collected information from the library and internet. They recorded the data obtained after observing pond water in table 2.1 and the data obtained from observing soil suspension in table 2.2. They identified the microorganisms at protozoa, algae, fungi and bacteria. You can also conduct the same activity, children, and record if you find any of these categories of organisms. Like this, okay. This is the table you can see here. Now, did you also observe any of these microorganisms or something different? Record in your notebook and discuss in your class. In table 2.1 and 2.2, you have identified a variety of microorganisms. They are everywhere and we can only see them with a microscope. A device that magnifies them 100 to 400 times. Though microorganisms are small in size, they play an important role in our lives. So children, have you ever heard of viruses are microscopic and acellular? Viruses multiply when they enter a living cell. They may infect plant, animals or bacterial cells and may cause a disease. How are we connected to microbes? Let us see. Can we find microorganisms in other places too? Let's have a discussion. Have you ever seen a lemon, tomato, orange or any other food item rot after being left outside for some time? If yes, you may have noticed a powdery or cotton-like growth on them like this. This happens because they have been infected by microbes. But where did these microbes come from? How did they come in contact with the food? This happens because microorganisms can be found everywhere, be it in water, soil, air, or even in some food items. Okay? So you can use a microscope to explore surfaces of leaves, stems, roots, or any other part to see them. Like plants and animals, microorganisms also show great diversity. Some of them can even be found in extreme climatic conditions such as hot water springs and snow cold zones, as well as at moderate temperature. You already know, children, that some of these organisms live inside our bodies, especially in our gut. You have studied in the chapter Life Processes in Animals in Curiosity grade 7 that our intestine has many bacteria that help in digestion like plants and animals. Microorganisms vary in shape, size and structure in table 2.1 and 2.2. You would have observed microorganisms of different shapes: spherical, rod shaped, or irregular. How does the diversity of microorganisms play a role in our daily life? How do they help clean the environment? So children, key players in cleaning the environment, let us attempt to understand this by doing an activity. Take an empty container and fill it halfway with garden soil. Add some fruits and vegetables peels to the container. Okay? Thereafter, put a layer of soil on it and leave it aside. After 2-3 weeks, observe the changes that have taken place. Do you observe any difference in the contents of the container? Okay. So, you may find that peels of fruits and vegetables have turned into a dark colored material. This is manure, which is rich in nutrients and helps increase the fertility of the soil. But how did the peels of fruits and vegetables turn into manure? In activity 2.6, you saw that soil contains various kinds of microorganisms. Remember, some of these microorganisms like fungi and bacteria act on the plant waste and slowly break it down into simpler nutrient-rich manure. You may have seen gardeners in your school or in a field near your house collecting dry leaves and plant waste and putting them into pits, right? Do you now understand why they do this? Yes, they are trying to make manure. It is to make natural manure. Now, our scientist heritage is here. Ancient Indian texts, particularly the Vedas, have references of the word krimi, which means different tiny entities, including drishya, that is visible, and adrishya, that is invisible. Various Vedic texts mention their beneficial and harmful effect. Okay? Atharva Veda also refers to krimi. If you look around carefully, children, you might see decaying plants and fallen leaves stored in a container or lying in the garden disappear after some time from the surroundings, right? How it happens? This is because microorganisms break down the complex substance of fallen leaves into simpler substances that are rich in nutrients, a process called decomposition. These nutrients go back to the soil and helps plants grow better, right? Microorganisms also decompose bodies of dead animals. So, microbes help recycle the waste and return important nutrients to nature. Manure formation occurs at optimal temperature and appropriate moisture level. Isn't it interesting, children? By now you must have understood that bacteria and some fungi are types of microorganisms that play an important role in our life. And guess what? These helpful bacteria can also decompose animal waste like dung. From activity 2.7, we can also infer that microorganisms not only help in plant growth but also clean our environment by breaking down waste. Now think what would have happened, children, if microorganisms did not exist on earth. See, there are many microorganisms like bacteria, fungi live in an oxygen-free environment. Some of these bacteria have the ability to decompose plant and animal waste present in the environment or household waste water. Okay? They don't need to live in an oxygen full environment. Okay? During the process, they release a mixture of gases called biogas, primarily composed of carbon dioxide and a high proportion of another gas, methane. This gas has been used as a fuel source for cooking, heating, generating electricity and to even run vehicles. So, we have a scientist here, Ananda Mohan Chakrabarti, 1938 to 2020, was a scientist who studied bacteria. In 1971, he developed a special bacterium that could break down oil spills, helping to clean the environment. His discovery received a patent in 1980. Patent is a copyright given to a person so that no one else can copy, use or sell his or her invention without permission. So, patent is required. His work showed how microorganisms could be used to solve environmental problems like pollution. He is remembered for his contributions to science and for protecting the environment using microbes. What are the other problems which you think can be solved with the help of microorganisms, children? Can you tell me? How does the diversity of microorganisms help in our kitchen? Let's know about it. Microorganisms and food. Let us try to understand by performing activities in the kitchen. You can also perform these activities in your kitchen also. Okay. So, activity 2.8, take two bowls A and B. In each take 200 grams of flour, atta or maida. Whichever you want you can take and add a pinch of sugar, right? Dough in bowl A, dough in bowl B. Now in bowl A, add a small amount of yeast powder and mix it well with the flour. In bowl B, do not add any yeast so that we can compare the result of two bowls. Knead the flour of the two bowls with warm water to make soft dough. Okay, you can see the dough here. Cover the dough with a damp cloth and keep it in a warm place. Observe both the bowl after 4 to 5 hours. Did you find any change in the volume, smell or texture of the dough? If not, leave the dough for some more time. After some time, you may notice that the dough in bowl A, where yeast was added, has risen slightly, become fluffy and has a different smell compared to the dough kneaded without yeast. Okay? Why does this happen, children? This happened because of bacteria present in yeast. What is the role of yeast? Why did we add sugar and warm water to the flour? Yeast is a type of microorganism. It belongs to a group of microorganisms called fungi. Yeast grows well in warm conditions. You may recall from chapter Life Processes in Animals in grade 7 that like other organisms, yeast also respires and breaks down food to release energy for their growth and carry out life processes. During this process, carbon dioxide is released, which forms bubbles that makes the dough soft and fluffy. Yeast also produces a small amount of alcohol during this process, which gives the dough a slightly different smell. Okay, so the smell is changed. This special property of yeast is used in the process of making breads, cakes and more. Okay. In addition to yeast, some bacteria such as lactobacillus help in fermentation of batter for making idli and dosa and dough for making bhatura. Okay. Now, activity 2.9 is here. Take two small glass bowls, label them A and B. Pour lukewarm milk in bowl A and cold milk in bowl B. Now add a small spoonful of curd to each bowl and mix well using a spoon. Cover both bowls. Keep bowl A in a warm place and bowl B in a cool place like a refrigerator for a few hours or overnight. Okay. Observe the changes in the glass bowls. Write your predictions and observation in table 2.4. So here are the predictions and observations. Change in the appearance of milk, change in the color of milk, possible reason, right? So please do this activity, children. You will observe that in bowl A, the milk has turned into curd. You eat curd, it becomes curd with the help of bacteria. After a few hours and has become little sour. Whereas in bowl B, the milk has not curdled but it might be a little sour. Do you know why this happens, children? The curd contains several types of bacteria. One of them is lactobacillus. This bacterium feeds on the sugar in the milk, lactose, multiplies and ferments the milk to form curd. Instead of producing alcohol like yeast, these bacteria produce lactic acid which makes curd sour. Okay? These bacteria grow well in warm conditions. That is why curd is formed in bowl A but not in bowl B. We can categorize the microorganisms into different categories such as protozoa, fungi, bacteria, some algae and more. Some bacteria such as rhizobium form the swollen regions called nodules and live in them as shown in figure 2.12. Roots of certain legumes such as beans, peas and lentils have root nodules that contain rhizobium bacteria. These bacteria trap nitrogen from the air and make it useful for the plants. This helps plants grow better without chemical fertilizers. That is why farmers grow legumes in rotation with other crops. This naturally increases the nitrogen in the soil and keeps it healthy for the next crop. So this is the reason farmers grow legumes with other crops. Amazing microalgae, tiny helpers in water. Microalgae, children, are microscopic plant-like organisms that live in water, soil, air and even on trees. They make their own food using sunlight. While doing this, they also release oxygen and produce more than half of the earth's oxygen supply. They are rich in nutrients and serve as a food source for many aquatic animals. Some like spirulina, chlorella and diatoms are also used by humans as health supplements and medicines. Microalgae also help in cleaning water and are used to make biofuel. However, pollution, climate change and habitat destruction are threatening microalgae diversity and abundance. It is important to conserve these tiny organisms to protect the environment and maintain oxygen balance on Earth. Have you ever heard of, children, spirulina? A microalgae is called a superfood because of its health benefit. Spirulina is also a good source of vitamin B12, which is essential for our body. It is rich in protein, more than 60% of its body weight, and only a small amount of fat and sugar. Nowadays, farming of spirulina is becoming a feasible livelihood opportunity. You can grow spirulina easily by following these steps. Set a clear glass tank in a bright place away from direct sunlight. Cover the tank with a shade net and keep the tank at a place with moderate temperature conditions. Fill the tank with pond water. Add living spirulina collected from a pond. Stir the growing spirulina twice a week. After 3 to 6 weeks, spirulina may be harvested from the tank by filtering it through a fine cloth. Conservation of microalgae is a good practice for ensuring food security and livelihood development. Why is cell considered to be a basic unit of life? The body of all living organisms are made up of tiny building blocks called cells. Okay, now you know that. A single cell contains various components that helps organisms perform various functions. The bodies of all plants and animals are made up of many cells. Therefore, they are called multicellular, many-celled organisms. In multicellular organisms, cells carry out specialized functions individually but also cooperate with each other to increase the chance of survival. Some microorganisms such as bacteria and protozoa are made up of just one cell. These are called unicellular, single-celled organisms. They carry out all the functions necessary for their survival in a single cell. Other microbes like algae and fungi are made up of one or more cells. For example, yeast has only one cell and mold has one or more cells. Okay? Like animal and plant cells, the cell of microorganisms are also surrounded by a cell membrane. Cells of fungi additionally have a cell wall, but they do not have chloroplasts, so they cannot make their own food through photosynthesis. Bacteria do not have a well-defined nucleus and nuclear membrane. Instead, they have a nucleoid. This feature distinguishes them from cells of yeast, protozoa, algae, fungi, plants and animals. We have only looked at a few basic cell structures here, right? The cell has other components about which you will learn in higher classes, children. For observing subcellular components, we need microscopes with high magnification. An electron microscope magnifies the cell about 10 lakh times, where we can see more structures present in a cell. By now, you must have understood that all living beings, including microorganisms, are made up of one or more cells. Their cells differ in size, shape and structure. Plant and animal cells also have some differences, right? Understanding these differences help us learn how these organisms function differently. In this chapter, we have learnt about the beneficial microorganisms. However, there are some microbes that cause diseases in plants and animals, including humans. We will learn about some of the diseases caused by microbes in the next chapter. So, we have some snapshots related to your chapter, children. Let's read it. Microorganisms are small sized organisms and are not visible to the unaided eye. What we need to see them? Microscopes. They can live in all kinds of environments and even in the bodies of plants and animals. They are either unicellular or multicellular. Bacteria and protozoans are unicellular. Fungi can be unicellular or multicellular, while plants and animals are multicellular. The cell is a basic unit of life. The body of all living organisms is made up of cells. A cell contains various components which help the organisms perform their functions and survive. A typical cell is bounded by a cell membrane filled with cytoplasm and contains a nucleus. Plant, fungal and bacterial cells have an extra covering called a cell wall around the cell membrane. Bacteria lack a well-defined nucleus. Cells differ in shape and size. Their shape is related to the function performed by them. Bacteria, fungi and protozoa are different kind of microorganisms. Viruses are also small in size, but they are different from other microorganisms since they reproduce only inside the host organism. Microorganisms can be beneficial or harmful to us. Some microorganisms decompose the plant and animal waste into simple substances and clean up the environment. Some microorganisms reside in the root nodules of legumes such as peas, beans and lentils. They trap nitrogen from the air and increase the soil fertility. Yeast are fungi which are used in the process of making breads, cakes, pastries, idlis, dosas and bhaturas. Lactobacillus is used in the curd formation at home and fermentation process in food industry. Now, keep the curiosity alive, children. There are some questions. Various parts of a cell are given below. Write them in the appropriate places in the following diagram. So, you have this diagram here and these are the parts of cell. You will write it here at the right place. Now, we have second question, children. Anandi took 2 test tubes and marked them A and B. She put 2 spoonfuls of sugar solution in each of the test tube. In test tube B, she added a spoonful of yeast. Then she attached 2 incompletely inflated balloons to the mouth of each test tube. She kept the setup in a warm place away from sunlight. So, now what can be a possible explanation for this? So, you have given 4 options and the correct option is yeast produced a gas inside the test tube B which inflated the balloon. Okay. So, option C will be correct option. Now, she took another test tube, 1/4 filled with lime water. She removed the balloon from test tube B in such a manner that the gas inside the balloon did not escape. She attached the balloon to the test tube with lime water and shook it well. What do you think she wants to find out? She wants to find out whether the gas produced by yeast is carbon dioxide or not. Question number 3. A farmer was planting wheat crops in his field. He added nitrogen rich fertilizers to the soil to get a good yield of crops. In the neighboring field, another farmer was growing bean crops, but she preferred not to add nitrogen fertilizers to get healthy crops. Can you think of the reasons? Yes, the reason was, children, that beans have a natural nitrogen rich fertilizers, so there is no need to add nitrogen fertilizers when he was growing bean crops. Okay. Snehal dug 2 pits A and B in her garden. In pit A, she put fruits and vegetables peels and mixed it with dried leaves. In pit B, she dumped the same kind of waste without mixing it with dry leaves. She covered both the pits with soil and observed after 3 weeks. What is she trying to test? She is trying to test that whether the mixture of fruits, vegetable peels and dried leaves is able to make better compost rather than using only same kind of waste without mixing dried leaves. Question number 5. Identify the following microorganisms. I live in every kind of environment and inside your gut. I make bread and cakes soft and fluffy. I live in the roots of pulse crops and provide nutrients for their growth. So please answer this yourself. Question number 6 is design an experiment to test that microorganisms need optimal temperature, air and moisture for their growth. Okay. Now question number 7, take 2 slices of bread, place one slice in a plate near the sink. Place the other slice in the refrigerator. Compare after 3 days. Note your observations. Give reasons for your observations. So please do this experiment also. A student observes that when curd is left out for a day, it becomes more sour. What can be 2 possible explanations for this observation, children? Please think and tell me yourself. Now, observe the setup given in figure 2.15 and answer the following questions. You will observe this. What happens to the sugar solution in flask A? What do you observe in test tube B after 4 hours? Why do you think this happened? And what would happen if yeast was not added in flask A? Okay. So, please observe it and answer me. Now we have discover, design and debate page, children. Please read out these topics and try to solve them yourself.